ಸ್ನೇಹಿತರೆ, ನನ್ನ ಪರಿಚಯ ನಿಮಗೆಲ್ಲ ಇದೆ ಅಂತ ನಾನು ತಿಳ್ಕೊಂಡಿದ್ದೀನಿ. ಡಾಕ್ಟರ್ KS ಕುಮಾರ್ ಅಂತ ಸಿದ್ಧಾರ್ಥ ಪ್ರಥಮ ದರ್ಜೆ ಕಾಲೇಜಿನಲ್ಲಿ ಅಸೋಸಿಯೇಟ್ ಪ್ರೊಫೆಸರ್ ಆಗಿ ಕೆಲಸ ಮಾಡ್ತಾ ಇದ್ದೀನಿ, ಕೆಮಿಸ್ಟ್ರಿ. ನನ್ನ ಕೆಲವು ವೀಡಿಯೋಸ್ನ ಯೂಟ್ಯೂಬ್ನಲ್ಲಿ ನೀವು ನೋಡಿರ್ಬೋದು. ಇವತ್ತು ನಾನು ಮೆಟಬಾಲಿಸಮ್ ಚಾಪ್ಟರು, ಅಂದರೆ ಫೈನಲ್ BSc ಇರೋ ಬಯೋಕೆಮಿಸ್ಟ್ರಿ ಟಾಪಿಕ್ನಲ್ಲಿ ಒಂದು ಟಾಪಿಕ್ ಇದು. ಈ ಮೆಟಬಾಲಿಸಮ್ ಚಾಪ್ಟರ್ ಬಗ್ಗೆ ನಿಮ್ಮ ಜೊತೆ ಡಿಸ್ಕಸ್ ಮಾಡ್ತೀನಿ. ಈ ಚಾಪ್ಟರ್ ಡಿಸ್ಕಸ್ ಮಾಡುವಾಗ ನಾನೇನು ಮಾಡ್ತೀನಿ, ಅದಕ್ಕೆ ಸಂಬಂಧಪಟ್ಟಂಥ ಪ್ರಶ್ನೆಗಳನ್ನ ಅಲ್ಲೇ ಹೇಳ್ಕೊತಾ ಹೋಗ್ತೀನಿ. ಈ ಮೆಟಬಾಲಿಸಮ್ ಚಾಪ್ಟರ್ನ 2 ಭಾಗದಲ್ಲಿ ಕಂಪ್ಲೀಟ್ ಮಾಡಿದ್ದೀನಿ. ಇದು ಮೆಟಬಾಲಿಸಮ್ 1 ಅಂತೇಳಿ. ಇದರಲ್ಲಿ ನಿಮಗೆ ಮೆಟಬಾಲಿಸಮ್ ಅಂದ್ರೇನು, ಅನಬೋಲಿಸಮ್ ಅಂದ್ರೇನು, ಕೆಟಬಾಲಿಸಮ್ ಅಂದ್ರೇನು, ದೆನ್ ಸ್ಟೇಜಸ್ ಆಫ್ ಮೆಟಬಾಲಿಸಮ್ ಏನು, ಅದರಲ್ಲಿ ಪರ್ಟಿಕ್ಯುಲರ್ ಆಗಿ ಗ್ಲೈಕೋಲಿಸಿಸ್ ಅಂದ್ರೇನು, ದೆನ್ ವಾಟ್ ಈಸ್ ದಿ bridge reaction between glycolysis and the Krebs cycle, and what is the fate of the pyruvate under anaerobic condition. ಸೊ ಈ ಥರದ್ದೆಲ್ಲ ಕೆಲವು ರಿಯಾಕ್ಷನ್ಸ್ನ ನಿಮ್ಮ ಜೊತೆ ಡಿಸ್ಕಸ್ ಮಾಡ್ತೀನಿ. ಬನ್ನಿ ಸ್ಲೈಡ್ಸ್ಗೆ ಹೋಗೋಣ. ನೋಡಿ, ಈ ಸ್ಲೈಡ್ಸನ್ನು ನಾನು ನಿಮಗೆ ಪವರ್ ಪಾಯಿಂಟ್ನಲ್ಲಿ ಪ್ರೆಸೆಂಟ್ ಮಾಡ್ತಾ ಇದ್ದೀನಿ. ಇದು ಡಾಕ್ಯುಮೆಂಟಲ್ಲಿ, ಅಂದರೆ ವರ್ಡ್ ಡಾಕ್ಯುಮೆಂಟಲ್ಲಿ ಕೂಡ ನನ್ನ ಹತ್ರ ಸಿಗುತ್ತೆ. ನಾನು ನನ್ನ ಇಮೇಲ್ ID ಕೂಡ ನಾನು ಫಸ್ಟ್ ಸ್ಲೈಡ್ನಲ್ಲಿ ಕೊಟ್ಟಿದ್ದೀನಿ. ಸೊ ನೀವು ಯಾರಾದರೂ ಡಾಕ್ಯುಮೆಂಟ್ ಬೇಕಾದ್ರೆ ನನಗೆ ಇಮೇಲ್ ಮಾಡಿದ್ರೆ I will send the Word document also. Thank you. Let us see metabolism. ಓಕೆ ಸ್ನೇಹಿತರೆ, ನನ್ನ ಫೋನ್ ನಂಬರ್ ನಿಮಗೆ ಸಿಗುತ್ತೆ 9448447137. ಹಾಗೆ ನನ್ನ ಇಮೇಲ್ ID ಕೂಡ ಕೊಟ್ಟಿದ್ದೀನಿ. ನಿಮಗೆ ಏನಾದ್ರೂ ವರ್ಡ್ನಲ್ಲಿ ಡಾಕ್ಯುಮೆಂಟ್ ನೋಟ್ಸ್ ಬೇಕಂದರೆ ಈ ಚಾಪ್ಟರ್ಗೆ ಸಂಬಂಧಪಟ್ಟಾಗ ನಾನು ಕೇಳ್ಬೋದು. Let us see the metabolism. What is exactly the metabolism? Metabolism is the term for the set of chemical reactions that occur in the cell of living organism to sustain the life. In other words, you can say metabolism is sum of all the chemical reaction that takes place in cell. Of course, the metabolic process lead to growth and reproduction and allow living organism to maintain the structure and respond to the surrounding environment. So metabolism is all the chemical reaction that takes place in the cell. And let us see what are the different types of metabolism. Metabolism is of 2 types. One is an anabolism, other is the catabolism. Anabolism is the constructive process, whereas catabolism is a destructive process. So anabolism or biosynthesis is the set of biochemical reaction that construct the molecules from the smaller components. That is, usually these anabolic reactions are endergonic. That means they are all energy requiring and they are not spontaneous. And anemonic reactions are those that build complex molecules from the simple molecules. For example, the construction of the polymers and the growth of tissues and repair damage are the important anabolic process that takes place in the cells. And let us see some of the examples for this anabolic process. We are all familiar with this reaction, that is glycerol that reacts with fatty acid to make the lipids. So this is the glycerol unit, and it is going to react with the fatty acid and it is going to construct or build the lipids. And similarly, so we know that the construction of the sucrose molecule, the formation of the sucrose molecule to give the from the glucose and the fructose molecules. These are the examples. So in a simple way, you can understand that anabolism is a constructive process. It's endergonic reaction and it takes place in cell, and in this smaller units are joined together to give the bigger units. Let us see what is catabolism. Catabolism is quite opposite to the anabolic process. That is, it is nothing but the breaking down of the simple molecules, of course bigger molecules, to smaller molecules. So thermodynamically, these are the favorable reactions, and thermodynamically they are spontaneous in nature, of course. So these cells use this catabolic process to to generate the energy or to fuel the anabolism process. The catabolism, of course, it's an exergonic and it releases heat. For example, so catabolism of proteins, lipids, nucleic acids, and polysaccharides generates the amino acids, fatty acids, nucleotides, and monosaccharides respectively. That here the bigger molecules, that are bigger polymer units like this, and they are going to degrade to give their building units. And some more examples for this catabolic process, and of course they are the reverse of the anabolic process, and they are used to generate the energy for anabolism process. So of course they release small molecules for other purposes, of course. Next, some of the typical examples for this metabolic process you can notice here, that is the degradation or oxidation of the glucose molecule. So oxidation of the glucose molecule so lead to the formation of the carbon dioxide and water. Similarly, there is another example, that is the decomposition of the hydrogen peroxide also. So to summarize, catabolism is a destructive process. It is opposite to the anabolism. Catabolism is an exergonic process. The energy of the catabolic reactions is used to run the endergonic reaction, that is the anabolic reactions. Let us see next one. What are the stages of the catabolism? So stages of the metabolism, or you can say catabolism process in specific. We know that our food is a combination of the proteins, polysaccharides, and the lipids. So that is, our food consists of these three important components. In the first stage, and of course we know that these proteins are polymers and polysaccharides are the polymers, and lipids also are the compounds formed by the joining of the glycerol and the fatty acid molecule. In the first stage of the metabolism, these polymer units are converted to their building units. That is, proteins are converted into the amino acids, whereas the polysaccharides are converted into their monosaccharides, that is the glucose, fructose, galactose, like that, and lipids are converted into the glycerol and the fatty acid. The first stage of the metabolism is nothing but the conversion of the polymer unit to their building units. The second stage is the conversion of these building units, these line of compounds, to a common intermediate called acetyl coenzyme A. That is, the amino acids are converted into the acetyl coenzyme A or pyruvate, and of course the glycerol molecule is converted into the pyruvate or the acetyl coenzyme A, and the monosaccharide units are converted into the acetyl coenzyme A through the formation of the pyruvate. So totally, the second stage is nothing but the conversion of all these three monosaccharide units to a common intermediate called acetyl coenzyme A. In the third stage, that takes place in the presence of the oxygen. So in the third stage, this acetyl coenzyme A enters into citric acid cycle and loses carbon dioxide, and meanwhile it produces the energy. So totally we can say metabolism involves three steps. The first step is the conversion of the polysaccharide to the monomer units, and the second step is the conversion of the monomer unit to a common intermediate called acetyl-CoA, and third step is the conversion of the acetyl-CoA in citric acid cycle to carbon dioxide. Next. Let us see what is glycolysis. So glycolysis is nothing but the anaerobic degradation of glucose to pyruvate. And of course, it is a primitive pathway where even now the microorganisms retain this pathway to get the energy. This glycolysis is also known as the EMP pathway, that is embedded Merov and Parnas pathway, because these are the three scientists who have sequenced the reactions of the glycolysis. So in glycolysis, a 6-membered glucose is converted into a 3-membered pyruvate molecule. Of course, in the process, so many enzymes have participated, and these enzymes convert in a stepwise manner the glucose to a pyruvate molecule. Okay, so of course it enters the usually the in this case these are things which takes place in the cytosol. Of course, all of the glycolytic enzymes are found in this cytosol. See here, this is the overall reaction of the glycolysis. You can find that glucose unit with the help of the NAD molecule and the ADP molecule gets converted into pyruvate unit. This is the pyruvate unit, and meanwhile two ADP molecules are converted to two ATP molecule, and NAD plus is reduced to NADH and the H plus. This is the overall reaction of the glycolysis. Here you can observe that. So the glucose molecule is converted into the pyruvate molecule. So glycolysis is nothing but the conversion of glucose to pyruvate. And of course, in this process, say a 6-membered glucose molecule is converted into a 3-membered pyruvate molecule, 2 number of the pyruvate molecule. This involves the 10 reactions, and of course they are all supported by the enzymes. And this involves the 2 stages. In the first stage, the glucose is converted into 3-carbon dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, and in the second stage that is converted into the pyruvate molecule. Let us see one by one how this glucose is converted into pyruvate. The first reaction of the glycolysis is nothing but the conversion of glucose molecule to the glucose 6-phosphate. You can observe in this chemical reaction, this reaction is catalyzed by the hexokinase enzyme. And this reaction, magnesium ion acts as an activator here. This reaction involves the ATP molecule. The phosphate molecule from the ATP is transferred to the 5th position of the glucose, and you will get the glucose 6-phosphate. Remember, so this is one of the reaction where you can find out the that is transferase enzyme, that is hexokinase enzyme. Next reaction. Here you can see how this glucose 6-phosphate is converted into fructose 6-phosphate, and this is catalyzed by the enzyme called phosphoglucose isomerase. Basically, this is isomerization reaction. These 2 molecules are the isomers, and it is catalyzed by the isomerase enzyme, that is phosphoglucose isomerase enzyme. You can observe here, this is a glucose molecule containing the phosphate group in the 5th position, and it is going to convert it into a fructose molecule having the phosphate group in the 5th itself. The next thing is, of course, how this fructose 6-phosphate is converted into fructose 1,6-diphosphate. Again, similar to the first reaction, this reaction also consumes the ATP molecule. This ATP is converted into ADP and, of course, the inorganic phosphate. And here you can see one of the phosphate group from the ATP is transferred to this position, and you will get the 1,6-diphosphate. And of course, the enzyme is phosphofructokinase. Again, it is supported by the presence of the Mg2 plus ion. The first reaction and the third reactions are the reactions where the ATPs are utilized in glycolysis process. Then we shall proceed to the next reaction. And the fourth reaction, it is the end of the first stage of the glycolysis. Here you can see that a diphosphate, that is fructose 1,6-diphosphate. It is a 6-carbon species, splits like this with the help of the enzyme called aldolase, leading to the formation of dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So dihydroxyacetone is a 3-carbon species, and glyceraldehyde 3-phosphate is again a 3-carbon species. You can observe, so two types of molecules. Of course, these are the isomers. In this next reaction, these dihydroxyacetone phosphate is converted into glyceraldehyde 3-phosphate. And of course, for that, the enzyme phosphate isomerase is involved. So fourth reaction is catalyzed by the aldolase enzyme. So where the 6-carbon fructose 1,6-diphosphate is converted into two molecules, that is dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, which are having the 3 carbons each. And of course, these two compounds are interconvertible to one another. Next, we shall see how this triose phosphate isomerization takes place. So again, that is dihydroxyacetone phosphate is converted into glyceraldehyde 3-phosphate and triose phosphate isomerase is involved. Next, we shall go for the next reaction, that is where the glyceraldehyde 3-phosphate is converted into 1,3-diphosphoglyceride. You can closely observe the structure, that is the phosphate group is there in the third position, and this phosphate group with the help of the NAD gets converted and accumulates one more phosphate group, and that is converted into 1,3-diphosphoglyceride. And here the NAD plus is converted into NADH and H plus. You can observe that NAD plus is actually reduced to NADH and H plus in this process. So you can notice here the phosphate group is joined in this reaction. And of course, the enzyme is called glyceraldehyde 3-phosphate dehydrogenase, that is GAPDH. The next reaction is again the conversion of 1,3-diphosphoglycerate to 3-phosphoglyceride. It's very important reaction where this 1,3-diphosphoglycerate is converted to 3-phosphoglycerate, and this inorganic, that is ADP, is converted into ATP. Here the production of the ATP begins. One of the phosphate group, that is from the first position, is transferred to the ADP, and the ADP becomes the ATP. In this reaction, ATP is produced, and of course in the first stage 2 ATPs are consumed, and here the production of the ATP per molecule of 1,3-diphosphoglycerate is actually shown. In the next reaction, that is 3-phosphoglycerate with the help of the phosphoglycerate mutase is converted into a 2-phosphoglycerate. You can see there is a phosphate group is in the third position. There it is shifted to the second position. The enzyme is that mutase, and that is phosphoglycerate mutase. So then the important reaction, that is which is catalyzed by the enolase, where this 2-phosphoglycerate is converted into phosphoenol pyruvate. That is 2-phosphoglycerate is converted to phosphoenol pyruvate. You can observe this phosphate group is transferred from this group to the second position. You can find out this compound 2-phosphoglycerate is converted into a double bonded one. It is going to lose water molecule OH from here and H from that, and it is going to lose the water molecule, and this is the enzyme involved in the conversion of the 2-phosphoglycerate to phosphoenol pyruvate, that is shortly known as the PEP. The next and the last reaction of the glycolysis is the conversion of the phosphoenol pyruvate to pyruvate. It's a last reaction. You can notice that phosphoenol pyruvate, after transferring this phosphate group to the ADP, which is going to become the ATP, gets converted into pyruvate. So this is the last reaction of the glycolysis. Here also you can observe there is a production of the ATP molecule. Remember, in the first stage, in the first reaction as well as in the third reaction, two ATPs are consumed. Whereas in the second stage, there is a production of ATP molecules in the two reactions. So each glucose molecule in the first stage is converted into two number of 3 carbon species, that is dihydroxyacetone phosphate and the glyceraldehyde 3 phosphate. Dihydroxyacetone phosphate is a 3 carbon one, glyceraldehyde 3 phosphate is also a 3 carbon one. They are interconverted to each other. And of course, each 3 carbon unit is 6 carbon unit is converted into a 2 number of the 2 carbon unit. Therefore, this is converted into 2 number of ATPs are converted into 4 number of ATPs. In this reaction, what exactly your question in your examination? You remember, so all the 10 reactions are important, and you may ask to write any 2 reactions. In glycolysis, how glucose is converted to glucose 6 phosphate, or how glucose is converted into fructose 6 phosphate, how fructose 6 phosphate is converted to fructose 1,6 diphosphate. Any of the reaction may be asked. That is asking the reactants, how they are converted. These reactions can also be asked in a different way. That is, what is the reaction catalyzed by the enolase? What is the reaction catalyzed by the enolase? What is the reaction catalyzed by the phosphoglycerate mutase? By name of the enzyme also, you may be asked to write the equation. In another way, so what is the reaction that is going to consume the ATP molecule? What is the reaction that consumes the ATP molecule? That is the first reaction and the third reaction. That is, phosphorylation process consumes the ATP molecule. So what are the reactions in which the ATPs are produced? When 1,3-diphosphoglycerate is converted to 3-phosphoglycerate, there is a production of the ATP molecule. Under the last reaction, that is when the phosphate group from the PEP is transferred to ADP molecule to generate the pyruvate molecule. So ATP involving reactions. Okay. So in this way also the reaction may be asked. So either in the name of the reactant, or in the name of the enzyme, or in the name of the participation of the other reactant. There is another possibility. In one of the reaction, that is, I will show that reaction. In this reaction, NAD plus is reduced to NADH. They may ask you to write, give the reaction of the glycolysis in which the NAD plus is reduced to NADH. In that way also the reaction may be asked regarding the glycolysis. So this is the overall presentation of the glycolysis, where the glucose molecule is converted into the pyruvate molecule, where a 6 carbon glucose molecule is converted into a 3 carbon pyruvate molecule, and it involves the 10 chemical reaction. It involves 10 enzymes. It involves 2 stages. So in the first stage, ATPs are consumed. In the second stage, ATPs are produced. Remember, glycolysis is an anaerobic degradation of glucose to pyruvate. Next, we shall go to the energetics of the glycolysis, or ATP yield during the glycolysis. I already told about the consumption and production of the ATP molecule. In the step 1 and 3, there is a production of the 2 ATP, that is the utilization of the 2 ATP molecule. In the 7th reaction and the 10th reaction, the 2 ATPs in each reactions are produced. So that is very simple. The net visible ATP produced per molecule of glucose during this glycolysis is the 2 ATP. For every conversion of glucose to pyruvate, there is a production of the 2 ATP molecule. So relatively, this energy is very, very small. But this small amount of energy is sufficient to carry out the cellular activities in the microorganisms. Then we shall see what is the fate of this pyruvate? What is the fate of the pyruvate? Suppose if the pyruvate continues to stay in the anaerobic condition, what are the possibilities? Suppose if it is exposed to the aerobic metabolism or aerobic condition, then what is the future of this pyruvate? You can notice here I have mentioned the 3 points. In the presence of the oxygen, that is the aerobic condition, so the pyruvate enters into tricarboxyl TCA cycle, where it is converted into the carbon dioxide. So that, and it is going to yield the large amount of the energy. So if it is the aerobic one, it enters into the tricarboxylic acid or TCA cycle. There it loses the carbon dioxide. There it releases the large amount of the energy. And most important here is the anaerobic condition. What is the fate of the pyruvate under the anaerobic condition? So in anaerobic condition, it results in the formation of lactic acid with the help of the enzyme called lactate dehydrogenase. There are two possibilities. With the help of the lactate dehydrogenase, the pyruvate is converted into lactic acid. So another one is the ethanol fermentation. That is, with the help of the pyruvate decarboxylase, it gets converted into with the help of the pyruvate decarboxylase and alcohol dehydrogenase, it is going to form the ethyl alcohol, and that is called as the alcohol fermentation or ethanol fermentation. So in short, we can say under the aerobic condition, it enters into the TCA cycle. Under an aerobic condition, either it may be converted into lactic acid or it may be converted into ethanol. The enzymes involved in the lactic acid formation of the lactate dehydrogenase are enzymes involved in this conversion with the alcohol fermentation is the pyruvate decarboxylase and alcohol dehydrogenase. Let us see the reactions of this pyruvate, feta pyruvate reaction. That is, these are the reactions of the fate of the pyruvate under the different conditions. You can observe in the first reaction the pyruvate to lactate. Lactate dehydrogenase is involved. The pyruvate molecule is converted into the lactate molecule. So lactate dehydrogenase, of course, and is slightly reversible, and in this process NADH is reoxidized to NAD+. Most important thing, under an anaerobic condition, in the glycolysis reaction, NAD+ is reduced to NADH. Whereas in the same anaerobic condition, that NADH which is reduced here is reoxidized to NAD+. And this pyruvate, with the help of this enzyme, converted into lactate. This is one possibility. Another possibility here is the pyruvate, with the help of the pyruvate decarboxylate, gets converted into acetaldehyde. And of course, decarboxylation takes place here. And this acetaldehyde, with the help of the NADH and alcohol dehydrogenase, gets converted into ethanol. The latter is called as the alcohol fermentation. To conclude, the fate of pyruvate under anaerobic condition is either the conversion of pyruvate to a lactate or the conversion of pyruvate to ethanol. Pyruvate to lactate involves the participation of the that is lactate dehydrogenase enzyme, and the conversion of the pyruvate to ethanol involves the two enzymes, that is pyruvate decarboxylase and alcohol dehydrogenase. And this is also asked in the examination, what is the fate of pyruvate under anaerobic condition, or what is the how the pyruvate is converted into the pyruvate is converted into lactate in anaerobic condition, or how the pyruvate is converted into ethanol in the anaerobic condition. Let us go for the next thing, that is the aerobic metabolism. Before going to aerobic metabolism, all these things are the anaerobic things. Anaerobic, and under the anaerobic condition, these conversions will be there. Okay, to conclude my talk, particularly that is the aerobic metabolism of glucose. Or also it can be noticed as the bridge between the glycolysis and the Krebs cycle. So the last step of the glycolysis, that is a 3-carbon pyruvate molecule, so is with the help of the oxygen or the aerobic condition, in the first step gets converted to acetyl coenzyme A, acetyl CoA. A 3-carbon pyruvate is converted into a 2-carbon acetyl coenzyme A and loses the carbon dioxide molecule. This is a very, very complex reaction. Here there is a participation of the coenzyme ASH, and of course this coenzyme ASH is attached to this pyruvate molecule and forms the acetyl CoA. And this reaction is supported, it involves the participation of the NAD plus, where it is converted into NADH. And it involves the participation of the enzyme pyruvate dehydrogenase complex. This is called pyruvate dehydrogenase complex because many enzymes participates here, and of course it involves the TPP, it involves the lipoid, it involves the FAD. So that's why this is a very, very complex reaction. Remember, here there is a participation of the coenzyme ASH, there is a conversion of NAD plus to NADH, and there is a participation of a group of enzymes. It also involves the activators, that is the TPP, lipoid, and FAD. These are the electron carriers, and of course participated in this reaction. And this is a bridge reaction because pyruvate is the final product of the glycolysis, and acetyl coenzyme A is the beginning of the tricarboxylate, that is TCA cycle. Okay, so this also may be asked as it is in the examination. I am going to conclude this metabolism chapter, and the expected questions are there in this chapter. So you have to remember all those questions, and in the reactions we expect you to write the chemical equation in your answer. Then I'm going to upload one more video where it covers the reactions of the Krebs cycle and energetics of the Krebs cycle, and the metabolism of the fatty acid, activation of the fatty acid, reactions of the beta oxidation, and the protein metabolism, where the important phenomena like transamination and deamination process, decarboxylation process, etc. And I am going to give the energetics of the beta oxidation also. Expect the next video, and it is going to give you the information regarding the remaining content of this chapter. Thank you. And if you have any suggestions, you can mail me, you can telephone me, and you can give your suggestions, and you watch my videos in YouTube channel, Dr. KSK channel. Subscribe to this channel and give your comment and give likes. Thank you.